pleased to introduce Professor Guoping Feng, the last speaker of this event during the day. He joined the McGovern Institute in 2010 and holds the Poitras Professorship in Brain and Cognitive Sciences. He's also the Director of Model Systems and Neurobiology at the Stanley Center for Psychiatric Research and an institute member of the Broad Institute. He's played a pioneering role in our understanding of the brain at multiple levels, including how neurons communicate, and he's really led the field and continues to do so in developing models of disorders, including autism spectrum disorders and schizophrenia. His talk title, Thalamic Reticular Nucleus Dysfunction in Neurodevelopmental Disorders. Dr. Feng.
Thank you. I understand that I'm on your way to the reception, so I will try to be quick. First of all, I want to thank really Pat and Laurie McGovern for establishing this wonderful institute. I couldn't find a better place to work than, truly, better place than the McGovern Institute. But that doesn't mean you can treat me betterly. So, it's incredible. I feel like both Pat and Laurie has been incredibly supportive. The best story is when we want to start the marmoset project, we couldn't find any marmoset. And Laurie went out of the way to, we found some in Japan but couldn't get it here. So Laurie went all the way to have breakfast, lunch, dinner, whatever it takes with her friends who has jets, private jets that can ship the marmoset from Japan. And she found someone actually paid all the shipment from chartered plane from Japan. So now we have 250 marmoset in this building actually. So this is incredible. So I have the opposite feeling. Actually, when you say life is good, time goes really fast, I couldn't believe I've been here for 9 years. I thought I was only here for like a couple of years. So Pat and Laurie is unbelievable, and their family is incredibly supportive. I never felt once they look over my shoulder. On the other hand, Bob always look over my shoulder. Have you made any progress yet? I'm seriously, he comes every day asking, how about the marmoset project? Did you make it work? So I want to clear that. Pat and Laurie, they never do that, but Bob does that all the time. You can ask other people. So today I'm going to talk about some project in the lab which is still ongoing, and you probably, some of you might, okay, doesn't work. Okay, so some of you might know my lab is interested in the corticostriatal thalamo-cortical circuits, and especially their dysfunction in neurodevelopmental and psychiatric disorders. There are two reasons. Anne made a great introduction and mentioned many times that this is the highly conserved, at least the parts of the circuit is highly conserved between species. And also, she also mentioned that the dysfunction of the circuit being implicated in many neuropsychiatric disorders and neurological disorders. And our interest is to understand both at the molecular, cellular, and the circuit level how these circuits function related to behavior, and how this dysfunction of these circuits might contribute to some of the symptoms or behavior problems in neurodevelopmental and psychiatric disorders. And today I'm going to talk about one particular aspect of this circuit called Thalamic Reticular Nucleus. And this is a GABAergic inhibitory group of neurons which is surrounding around the thalamus, and they provide the main source of inhibition to the thalamus. And this circuit actually this part of circuits, which is thalamic reticular TRN, play a very important role in controlling the output of thalamo output to the cortex. So it has many of different functions. One of the main thing it does is function like a gatekeeper, because we receive all the sensory input all the time, but we don't react to them at all. Otherwise, we go crazy immediately, right? So one of the function of the TRN is to filter out all the information you don't want and amplify the ones you really want. So, for example, in a party, really noisy, there's no problem you talk to your friend and ignore everything else, right? But so that's a very focused signal amplification. However, you're also very flexible. If there's a fire alarm, suddenly you will hear it and you will run out. So these are the functions that TIN play a very important role. And TIN also play a very important role in generating of the sleep signals, which is very important for sleep rhythm and normal sleep patterns. And TIN dysfunction has been implicated in many neuropsychiatric disorders, including autism and schizophrenia. The reason we are interested in this is that we found not only theoretically this is very important, but we also found many of the risk genes for psychiatric disorders are also highly expressed in the TIN. You can see this is a mouse sagittal section. You can see this is TIN. You can see these two genes are very on the top of the list of GWAS risk genes for schizophrenia. These are the monogenic causative genes of autism spectrum disorder. And we actually started with patch D1. Today I'm going to talk a little bit about patch D1 because we use the patch D1 as an entry point to study the function and dysfunction of the TIN. The reason we choose that is patch D1 mutation actually is a monogenic cause of a neurodevelopmental disorder with autism spectrum features. It has many other problems. And patch D1 is, we found that early on, patch D1 is actually very highly expressed in the TIN and very low expression in other part of the brain. And later on, it's still highly expressed in TIN, but other parts of the brain are also expressed. And it's X-linked, so almost all the patients are male, boys. And the patch D1 actually, patient has shown a variety of symptoms, including, intellectual disability, sleeping disorder, ADHD, hypertonia, and aggression, and also ASD, autism spectrum disorder features. And so we thought this might be an entry point for us to understand not only TIN dysfunction, but also how TIN dysfunction contributed to some of the symptoms found in patients. So this work was a very close collaboration with my Classes lab in many aspects from the beginning to the end. So I want to acknowledge early. This is mostly done by one of my graduate students, Mike Wells, former graduate student, and Ralph, a postdoc in Mike's lab. So when we made a conditional knockout, we deleted the germline, and we, previously collaboration with Chris Moore and Mike Halassa and my lab showed that TIN is play a critical role in the generation of sleep spindles. So in the mice, we found that actually Mike's lab did in vivo recordings show that indeed in these mice the bursting rate of this TIN neurons are reduced and lead to reduced sleep spindles. And these mice also showed disrupted sleep. And you can see that although the total time may be different, maybe similar, but the bouts, the length of bouts of sleeping is much shorter in the knockout. It means they wake up a lot more times. And mice sleeping is very different from humans. We don't wake up every few minutes, but the mice do. So, but you can see there's even shorter sleeping bouts in the mice. Now, the bursting of the TIN neurons have been well studied. They are controlled by two types of channels. One is the T-type calcium channel, CAV3.3. The other is the SK2 calcium activated potassium channel. And you can see that previous studies show if you knock them out, either one out, you will have reduced the sleep spindles. And so we use slice physiology. We showed that indeed in these knockout mice, the rebound bursting is reduced, and however, when you look at the channel activities current, the calcium channel current is not affected, but the potassium channel, SK2 channel current is reduced, suggesting that the defect may come from the defect of the SK2 channels. Since the SK2 channel is calcium activated, and we look at whether there is a calcium defect in these TIN neurons, and use calcium imaging, dye imaging, we show that indeed the calcium concentration, free calcium concentration, is reduced in these knockout mice, suggesting there is a defect in the calcium homeostasis in these neurons. Maybe this is contributing to the SK channel reduce the current in this, because SK channel are calcium activated potassium channels. So since the TIN has been, as I mentioned earlier, TIN has been implicated in the noise filtering and attention, Mike's lab actually, we use the Mike's behavior method approach. Mike lab developed that test the attention of these mice. And basically, these mice can, we train the mice to detect the cues. And in the normal condition, these mice will do very well. Both Y-type and knockout do very well. And then you can add a distractor, whether it's a distractor cue, distractor, and the mice have to remember this is not the cue that get a reward. And you can see in the distraction, in the presence of the distractor, the mutant mice make a lot more errors, suggesting that they have a problem to filter out these noises compared to the real signals. Now, suggesting they really, lack of cannot focus or distinguish between the cues and the noises. And these mice also showed hyperactivity. And this is the open field. You can see in the whole 60-minute test, they are hyperactive. And most of the conditions of hyperactivity actually in mice can be treated with, amphetamines, which is psychostimulant used to treat ADHD. But these mice actually are not effective. You can see that it does not do anything, suggesting there may be a different mechanism in this type of ADHD like a behavior in these mice. And so in addition to this sleeping problem, attention, and hyperactivity, these mice have a whole bunch of other problems, which it's quite amazing to me that you can see this in a mouse model. For example, it has a learning disability, whether you use fear conditioning or use inhibitor avoidance test, and you can see these mice are actually defective in fear learning. And the mice are also very aggressive. They actually will, whether in the just handling them, you can tell. You don't have the genotype. You put your hand into the cage, you know which one is knockout, because it will come bite you. So Mike Wells had to wear mittens to catch these mice. And the mice also have motor problems, except all these are found actually in patient, except it does not have we call social behavior or repetitive behavior we see in autism spectrum disorder. So we are interested in see whether all these are coming from TIN dysfunction, because early on, the TIN is the major site of expression of this gene, and later on is expressed in many other areas. So we decide to try to make a knockout only deletes this gene in the TIN. Now, that time there was no TIN specific cree, so it made it really difficult. So we use adapted approach used by Bo Lee's lab actually at Cold Spring Harbor. Basically, there's a somatostatin cree, which is somatostatin is a subgroup of inhibiting neurons overlapping with the patch D1 in the TIN. But outside the TIN, actually, it does not overlap with the patch D1 expression. So to prove that, we actually made a knocking mice, GFP knocking the patch D1 locus to show that they do, because there's no good antibody patch D1, we show that besides the TIN, there's no significant overlapping of these two gene expression. So that if we cross this conditional knockout with the somatostatin cree, then you can see it knock out only in the TIN. This is the control in TIN, but it's not in the other brain areas. These neurons still express patch D1. So with this knockout, we show that in the mice, conditional knockout of TIN, we will see attention deficit, hyperactivity, and a sleeping problem. But there's no problems in fear learning, in aggression, or motor activity, suggesting that the TIN defect is only responsible for the ADHD-like behavior or sleeping problems, but not all other problems. So we then test, is SK2 channel a real defect is the cause of this problem? And luckily there is a SK2 is SK channel activator called EBI 1-EBIO. So these can be used in mice, but in a lower dose. High dose is actually very toxic. What we found is that if we use this drug to IP injection to treat an animal, and in a knockout after 30 minutes, you can see that hyperactivity is reduced and attention deficit is corrected, but it does not correct the learning and other problems, suggesting that although SK2 channel defect maybe anywhere else, but the correct SK2 channel in the TRN maybe can correct the TRN related phenotypes. So the question to us now is, we got really interested, is there a better target than SK2 channel? Because SK2 channel is also in the heart and many other places you actually cannot use as a drug target. This compound is actually high dose, is toxic. And also in these symptoms we see in patients, including ADHD, attention deficit, hyperactivity, sleep problem, in many different disorders, and they are variable between patient to patient. Not everyone has the same thing. So how a TRN can such a small, seems uniform GABAergic neuron structure can control such complex different levels of behavior. So we want to see whether, first of all, whether there is a subnetwork kind of thing in the TRN control different function of the circuits, and the second, are there molecular targets we can identify to very specifically for TRN control, and maybe in the future we can develop a better tools or even better drugs to alleviate this kind of problems in neurodevelopmental disorders. So we decided to use a single-cell RNA-seq because we are here at MIT and Harvard. These are the inventors of this technology are all here. So we collaborate with several groups, including Zhang Yanfu's group at the Stanley Center, Joshua Levin's group at Stanley Broad, and Aviv's group at the Broad Institute. So first of all, we show that all these neurons are actually, TRN neurons are PV positive. So almost 98% of them, at least in mice, they are PV positive. They give us a way to identify these neurons. So we made a PV TD tomato mouse, so allow us to actually dissect on the slice, dissecting the TRN out, because the TD tomato red in the fluorescent microscope, you can dissect them out, then doing single-cell RNA-seq, and we can identify, of course, we know these are GABAergic neurons, and we know these are PV positive, GABAergic PV, so GAD positive and PV positive neurons are putative TRN neurons. So we want to see whether there are subclasses of cells in the TRN that can help us to distinguish different functions of these neurons, and hopefully that different connectivity. What we found was actually surprising. There are no clear clusters, and so it turned out that there are two opposite gradient kind of expression for each has hundreds of neurons, over 100 genes actually expression in a way that they are opposite. One group of one profile, one group of cells, you can see the expression are consistent with the one of the marker genes called SPP1. They express the most in this end, but much less expression in the other end, which is matched with another group of genes markers called ECEL1, that their gradient expression is the opposite of this gradient. So you basically have two group of cells that are with gradient expression, and at two extreme of these two groups, you can see this can be marked by SPP1, and there is no expression of ECEL1. But on the other hand, in extreme of this gradient, you see ECEL1 expression, but no SPP1. So we use these two genes as a marker to label the extreme of these two gradients to see whether they are molecular diversity, cellular functional diversity, connectivity diversity. Maybe these two cells can tell us something about the circuits function and the subnetwork in the TRN. What we found was very interesting that even anatomically, the expression, their location are very different. So this is the TRN, and you can see we use it's very hard to see here. We use PV for red and the purple, purplish for SPP1. Then ECA1 is green. What you can see is that the SPP1 neurons, positive neurons, are all in the middle, and the ECA1 neurons are at the edge. So this is like shell and a core kind of structure, even with this thin TRN structure, and because these two groups of neurons, they have very different profile gene expression, and many of the genes control the excitability, ion channels, and transporters. So we thought maybe electrophysiologically they will be very different. Indeed, when we look at this, we used basically we use the patch clamp technology to record the electrophysiology. After we're recording, we suck the content out doing single-cell RNA-seq to see what type of neurons this is. And what we found, we did many of the recordings and in different regions of the TRN to make sure we collected the whole areas. What we found is that the SPP1 neurons, they have multiple bursting. And within a bursting, they have much higher frequency of intra-bursting frequency. On the other hand, ECEL1 neurons, they have either single or zero bursts, so they're very low bursting frequencies. And within the bursting, also the bursting frequency is much lower. So they are rebound bursting property, which is the main feature of the TRN neuron, are very different from the two group neuron. Because ECEL1 is on the edge, so most of the time, if you don't know this, you're recording from, you want to make sure you poke the electrode in the middle so you don't miss the TRN. So that's why most of people, when we talk about it before, is always this rebound bursting TRN neurons. Actually, there is a group which on the edge of and they are actually have very few bursts. And also, if you look at the facilitations and the SPP1 neurons have facilitation, but ECEL1 neurons seems don't have a very significant facilitation. So they are suggesting their presynaptic input may be also very different. So these two groups of neurons not only molecularly defined differently, but they are functionally at a cellular level, they are also very different. And then we all know that in the TRN, in the thalamus, there are many different nuclei. At least, generally we can divide them into two type of thalamic nuclei. One is called first order, which is relayed of the peripheral sensory input into the thalamus, then go to cortex. The other is we call high order, which is basically connecting between cortical areas. Probably their function is more or less regulated. For example, multisensory integrations or other cortical interactions. And both of these group thalamic neurons receive TRN input. The question is whether they have do they have a differential input from the TRN control different function, because they are very different functions from this connectivity map. So to test that, we actually use retrograde beads tracing by injecting retrograde beads into either first order or high order nuclei of the thalamus, then look at where they are in the TRN. Right? So this is where you can say, okay, whether then you can do in situ and see, are these cells SPP1 positive or ECEL1 positive? What we found was very surprising. When we inject into the first order nuclei, all the retro beads go into the middle. And when you do in situ, these are SPP1 positives. And if you inject into the high order, and they all go to the edge, which is our ECEL1 positive, suggesting that not only molecularly, cellularly they're different, their connectivity is also very different, and they have very distinct connectivity between the TRN and different thalamic neurons. So we did multiple sensory areas. From each sensory area, you have first order and high order. They are all same. They are very same. That majority of high order receive ECEL1 positive neuron innervation from TRN, and the majority of the first order receive the SPP1 positive innervation. So in the TRN, although it's a very small thin structure, we have molecular gradient diversity in a form of molecular gradient, and they have different molecular electrophysiological properties. They also have, surprisingly, a very different connectivity. So that gives us an opportunity now doing in vivo functional study to see which of these circuits control what kind of behavior. And do they control, is the attention or sensory filtering different from the hyperactivity or other sleeping dysfunctions? And also allow us to do what are the molecular mechanisms, because we know every single gene now most of the genes expressing these cells, allows to really study what are the molecular mechanisms controlling the firing property, rebound bursting properties, or other properties in these neurons. So, and our hope is really use this information to identify subnetwork specific molecular target, allows to test and control or even modulate the different function, and hopefully this could in the future allows to develop, hopefully develop some kind of modulations, maybe applicable in the future in the clinic. And this also allows us to develop genetic tools. Now we can develop genetic tools very specifically to modulate the individual subnetworks. And for that, actually, Sasha Crow in the lab already developed some of this. Is developing using intersection method, developed actually TIN specific Cree lines. This is, you can see from the edge, so this is ECL1 Cree line. This is in the middle, you can see this is the SPP1 Cree line. So this will help us to really, now we can start to manipulate these different subnetwork in the TIN, look at how they control thalamocortical output and behavior. So I'm going to stop here. I want to acknowledge all the people who did this work. As I mentioned, Patrick Dewey is a collaboration between my lab and Mike Halassa's lab. And TIN, I mentioned, this was mostly done by 3 very talented people. Two postdoc fellows, 1 research science, associated at the Broad. Yingqing and Violata are both co-lead the project. Both got their faculty job. Yingqing is actually here, and he is a professor at Tsinghua University. And Violata just established her own lab in Mexico City. And Shen is a research associate in Joshua Levine's lab, and we have a lot of help from many different groups, especially with Zhang and Joshua and Aviv and Mike Halassa's lab for this TIN sequence. And many people in the lab actually helped in this project, and I'm also grateful for all the funding from the McGovern Institute, Simon Center, Poitras Center, and the Tanyang Center, and all other agencies. Thank you.